everyone. Uh, Welcome to this webinar that is hosted by Haystack Analytics. On behalf of my team at Haystack Analytics, I welcome you all and thank you for joining in. To begin with, we'll just start by giving you a brief overview of our organization and what we do, and then we will move on to the webinar for today. The topic for today's webinar is harnessing human genomic variation to advance disease diagnostics. When we talk about disease diagnostics, there are several aspects which we will try to cover today, as well as Dr. Jaya, who is the presenter today, will talk in detail about her work towards advancing disease diagnostics using genomics. To briefly tell you about what Haystack Analytics is and what we do. So Haystack Analytics is a health tech company. We are based out of IIT Bombay, and we believe in building scalable genomic solutions for the masses. We have developed several diagnostic tools which are based on completely genomics. This also includes our flagship product, which is the infection NGS product, which helps in diagnosis of more than 30,000 pathogens in 24 hours using a single diagnostic tests. This aligns with WHO's Sustainable Developmental Goals. In addition to infection, we also work on non-infectious diseases, including human genomics. Under human genomics, we have several products which include whole exome sequencing, pharmacogenomics, etc. Our journey has been started in 2019, just before COVID, when we won the Maharashtra Startup Award, which helped us to begin with our first work, that was diagnosis of TB using whole genome sequencing. Using this product, we were able to give not just the identification of the bacteria, but also NTMs that might be involved in TB, as well as drug susceptibility to about 18 antibiotics. So with that, we started our journey, and building over the last 6 to 7 years, we have come up with several different diagnostic solutions, for which we have also been appreciated by several government agencies, including BIRAC, as well as other governmental agencies. In 2023, we took a step ahead when we went from just TB diagnostics to launching the universal ID test. The proof of concept was achieved in 2023, and after that, we started working on this test, where, like I mentioned previously, we are able to detect bacterial and fungal infections as well as antimicrobial resistance in a single test, and that too within 24 hours. So building on this product, in 2024, we also won the FICCI Health Digital Transformation Initiative Award for that year. And 2025, we went global, where we won the TICON Award at Boston. We were the overall winners as well as the winners in life sciences. And more prestigious is what we achieved in 2025 is NABL accreditation of our Mumbai facility. So with that, I will wrap up the introduction of the company. To introduce to you today, the speaker for today, who is Dr. Jaya Srivastava. Jaya did her PhD from Indian Institute of Technology, Bombay. And after that, she has been working with the NIH for last several years. So her work focuses on uncovering the genetic pathways of non-coding regulatory variation in complex diseases. She uses deep learning as well as statistical modeling approaches to study the functional impact of different regulatory variants on human traits. She also her work also helps in understanding the ongoing accumulation of population level variants, which eventually shape, the phenotypes as well as influence susceptibility to neurodevelopmental disorders. She works with regulatory genomics of endocrine disorders and age-associated dementias. And she also focuses on how enhancer dysfunction and allele-specific regulatory effects contribute to disease mechanisms. Her long-term goals include elucidating how population-specific genetic variants influence disease risk and prevalence and advanced genetically informed diagnostics. So I think Jaya is a perfect speaker for us. Her thoughts and her way of working, as well as her research topic, aligns greatly with what we do at Haystack Analytics. So I'll hand it over to Jaya.
Thank you, Dr. Shailesh. Shailesh forgot to mention that we were colleagues at IIT Bombay and also bird watching buddies. I'm going
Yeah.
to share my screen.
Yes. Yeah.
I hope that's
Yes, we
visible.
can see it.
Okay.
Yeah.
Okay. Uh, I wanted to thank everyone who has joined to listen to this talk. And I think Haystack's analytics venture into whole genome sequencing is very exciting for some of the reasons that I'll mention in my presentation today. I'm going to talk about human genetic diseases and how these contemporary next generation sequencing technologies are going to help us in advancing our understanding of disease mechanisms and therapeutic strategies. Oops. Okay. So the human genome has about 3 billion base pairs, and only 2% of them are protein coding. But the rest are were used to be called junk DNA, but now we have a greater understanding of what they might be functioning. I'll talk about those regions in detail. But the genetic traits and disorders, by traits I mean the color of the eye, height, weight, etc., and disorders, they arise from variants in our genome. So this is a normal gene, and when there's a mutation in the gene or in a surrounding region that controls the expression of this gene, and I'll talk more about that controlling mechanism later, uh, may lead to either aberrant expression of the protein or levels of the protein. These mutations can either up or down regulate the gene expression that can lead to trait difference in traits across individuals or diseases. Um, so talking about diseases though, there can be two types. One is monogenic disorder, that is there can be mutations in just one gene that can lead to a phenotype. For example, cystic fibrosis, sickle cell anemia. And then there are polygenic disorders where these are multifactorial disorders, mutations in multiple genes, or they can lead to appearance of a phenotype like obesity, diabetes, cardiovascular diseases, etc. All of these variations in our genome have been cataloged in a database called DBSNP. This is hosted by the NIH. And typically, these IDs look like this. They start with, they are prefixed with RS, and there's a unique identifier. This particular variant is associated with a gene called GLP-1R, which is the target for all the GLP-1 based drugs. Now, uh, as I said, variation in gene expression doesn't only lead to appearance of disorders, it also leads to individual or species specific differences in traits. And for example, there is the large scale, so far, the largest scale of measuring such expression due to human genomic variants was carried out by the GTEx consortium. They collected 15,000 RNA-seq samples from about 800 postmortem donors from 49 tissues. And through statistical association, they correlated the variation among these individuals to variation in gene expression. And the ones with significant association have been cataloged in their database, the GTEx portal. An example here is with respect to a variant that's associated with Alzheimer's disease. This is a variant that's associated with the gene called BIN1. And the expression of this gene varies across different tissues with either the homozygous or heterozygous state of the allele that's present at this with this variant. Shown here are different levels of gene expression in blood, pituitary gland, and heart. Um, so, by measuring how gene regulation and expression is controlled by these genetic variations, will vastly expand our understanding of molecular origins of different race and diseases. And I think whole genome sequencing is perhaps the best effort towards this cause. So GTEx has so far cataloged about 4.3 million variants, but that's not all that's found in our genome. There have been several sequencing efforts, whole genome sequencing efforts by different consortiums like Thousand Genomes Database, EXAC, and more recently, and the largest of them all, called NOMAD, Genome Aggregation Database, that has sequenced zero point 0.7 million exomes, which uncover coding variants, and then 76,000 whole genome sequences. And in total, they have cataloged about 780 million single nucleotide variants. And this excludes the other structural variants or indels that are present in our genome. So we are talking about a huge catalog of genetic variations that are found across so many individuals. And these individuals belong to different ethnicities. Most of them are actually European. And I'll also talk about why overrepresentation of a particular cohort can be a bit misleading in interpreting diseases. But anyway, so a majority of these variants are either very rare or singletons. By singletons, I mean that those variations are found only in one individual. And mm, although most of them can be benign, they may have no effect on diseases, but some of them are disease associated. And the efforts go into understanding how these variants are associated with diseases. One of the things that these efforts uncover are a certain set of genes, and so far about 3,200 genes have been recognized as loss of function intolerant genes. These genes, among 20,000 genes that we have in our genome, are severely depleted of any of these variations, which means that if you have a variant, it's more likely to cause a disease. And I'll talk about those in a slide. That's my current project. But coming back to this database, so it's good that we have a huge catalog of human genetic variation, but how do you associate? So if you want to find out how these variations are associated with a phenotype or a disease, you need to know the profile of that individual from which the whole genome sequence was isolated, right? Which is currently not extensively characterized for Nomad and other databases. But then there have been many efforts across different countries uh, to perform something called genome-wide association studies. So this is where you associate variants in an individual to the corresponding phenotype. What they usually do, for example, let's say they want to find out variants that are associated with obesity. They take they take cases and controls. These are cohorts, patient cohorts and control cohorts, and they perform SNP microarrays on these cases and controls, and then identify those variants that are significantly more associated with cases than controls and these statistical associations eventually lead them to uh, the result is Jiva summary statistics, which typically looks like this. You have variants from across the genome present in cases and controls, pre- present in cases that some of these may be passing the significance threshold of these associations, and those are the loci, the genetic loci that are associated, that are believed to be associated with obesity, for example. Now, there's a huge catalog of such studies called the NHGRI ABI catalog, Jiva's catalog, where you have Jiva studies from thousands of traits and diseases, and it's free to browse. It's available for public use. But then I think in the past decade, biobanks came into picture. So the largest of them so far has been the UK Biobank, but there have been other efforts by, I think All of Us also is competing. All of Us is a whole genome sequencing effort in the United States, then the Million Veterans Program, Biobank of Japan, FinGen, etc. So what the advantage of these biobanks as opposed to Nomad genome variation aggregation database is that first of all, they have genotype arrays or whole genome sequencing technologies. So they have sequenced about 490,000 whole genome sequences, 454,000 whole exome sequences, and they have linked these to phenotypes by performing participant surveys on a range of topics like individual lifestyle, blood work, dietary factors, etc. And in doing so, they have paired the genetic information with phenotype information, and then they perform genome-wide association studies. So far for UK Biobank itself, we have variants that are associated with about 7,000 phenotypes, and other biobanks like FinGen from Finland, Estonian Biobank, etc. All of them have performed some kind of Jivas, and it's free to use. So the advantage of using these genome-wide association studies from other Jiva studies, which typically involve a very small cohort, it can be either hundreds or a few thousands, is that now these biobanks have a detailed data of not just one phenotype, but other traits and diseases that may be associated with an individual. So and these records are preserved through external data linkages like electronic health records. And I'm mentioning this because I think it will be very important in the Indian biomedical industry to have a thorough understanding and record of electronic health records so far if you want to extract meaningful data from the ongoing whole genome sequencing efforts. So once you have these broad range of phenotypes associated with an individual or a cohort, then you can ask questions like if you are studying, for example, PCOS, what kind of variants are associated with PCOS? You can also ask whether is BMI affecting PCOS? Is our sleep schedule affecting Alzheimer's disease? And things like that. That's a very interesting avenue for especially population-specific studies. So, but perhaps the most important information that came out of these whole genome and exome sequencing efforts was that, and Jiva studies, was that a majority of the genomic variants, they lie in the non-coding genome. And as I said earlier, only 2% of our genome codes for proteins, but the rest used to be called junk DNA. But after the human genome sequence was released in 2001, after that, there have been several efforts, particularly spearheaded by this consortium called ENCODE, where they started characterizing the function of these regions. And turns out that most of them, they are involved in some kind of gene regulation. Um, uh, and even all of these Jiva studies have also highlighted that 95 to 98% of the variants that are uncovered in these phenotype association studies, they lie in the non-coding genome. Non-coding genome can be regions like enhancers, promoters, even distal enhancers and silencers, etc. I'll talk a bit about those in detail in the next few slides. So, but a little about how the coding variants differ from non-coding variants. So the ones here in yellow, they are coding variants. They can be missense variants, synonymous variants, and especially if it's missense variant, it can be quite deleterious to the protein. It can alter the structure of the protein or the expression itself. But then there are these non-coding variants that can be present in splice sites, promoters, enhancers, etc. These lead to up or down regulation of gene expression mostly. So they can alter the dosage of the protein rather than the structure that is modified by the coding variants. Now, okay, so a little bit about these regulatory mechanisms. I already there are 20. I told you that there are 20,000, about 20,000 proteins, right? Not all of these proteins are expressed across more than 400 cell types that we have in our body. So these non-coding regulatory regions are responsible to maintain that status. They ensure that there is a spatiotemporal expression of genes, and this is a culmination of efforts by different factors. So the schematic shows a gene that's promoter of a gene, and then different factors. So our genome is actually in a silenced or repressed state when there is no expression. But when there is a need for expression in a particular cell type at a particular time point, then there are different factors like activated enhancers. There are histone modifications, opening of the DNA, and then removal of silenced regions. All of these different types of modifications on our histone proteins around which the DNA is bound, that leads to activation of that region. And then there are different TFs and their transcription factors and their cofactors that bind together to create a chromatin niche that becomes a playground for gene expression. And actually, these non-coding variants that I spoke about, they can lie in any of these they can be associated with any of these regulatory factors and lead to small changes in gene expression that can lead to trait differences like eye color, but they can be larger differences beyond a threshold that can also lead to a disease. But some of the very cardinal points about these regulatory regions is that, as I said earlier, these are they perform a cell type specific targeting of gene expression, and because that is the case, then there are different sets of regulatory regions active in different cell types that control the same gene's expression, and there can be multiple enhances silencers or enhances or silencers or other regions that can control the same gene expression, but they can be different regions in different cell types. Case in point, here is an intronic region that's an enhancer in the gene of intronic region of FTO. Now, this region in neurons and preadipocytes doesn't regulate the expression of the gene where it's contained, and not FTO. But in these two cell types, it's actually linked to expression of a neighboring gene called IRX3 and IRX5. These links, these are called chromatin contact domains. These are available from high-C data. That's a type of next generation sequencing technology. I'll talk about that a little bit later. The data shown here is for neurons and preadipocytes, but there are other cell types where the same region can also be involved in expression of FTO. Now, this is how cell types maintain spatiotemporal expression. And a property of these enhancers or silencers is that they skip genes quite often. Only in 50 to 80% of the cases, depending on the cell type, is the proximal gene the target gene. But sometimes in 50% of the cases, they can actually skip genes. Now, there are factors that lead them to do so, but I'm not going into the details of that right now, maybe in another webinar. But and I've convinced you that gene skipping is a very pervasive phenomenon among enhancers. So how do we identify these regions in our genome? First of all, because these are cell type specific, we need to identify these regions in a cell type specific manner. So there are sequencing technologies called ChIP-seq, DNA-seq. What they do is there are antibodies that are developed against these modifications, like HGK27AC to detect enhancer regions. Then there are chromatin accessibility assays as well called DNAs. Once these assays are performed, you get reads which are aligned to the reference genome. And then these reads will tell you where open chromatin is present. And when that overlaps with other regions like enhancer activity, it means that that's an active enhancer region in a particular cell type. So those profiles, these are called epigenomic profiles, and these are associated with one cell type. They look like this typically. There is this chromatin accessibility region. So this is a region in the genome in a cell type that's active, and coupled with other modifications like K27AC and K4ME1, which are marks of active enhancers. It means that this region is is controlling gene expression of its target gene. It can be a neighboring gene, and they typically overlap with some TF transcription factor binding sites. So if you have a variant present in this region, and these regions are like 200 to 400 base pairs. If you have a variant or multiple variants present in this region, they alter the binding of a transcription factor and lead to changes in gene expression. If this profile belongs to the pituitary gland, then you have changes here, but this profile may not be available in whole blood, in blood. So you may do, and there might be a different set of variants controlling the expression of this gene in blood. So the profile looks different. So, and another interesting thing about these regulatory regions is that so there was a paper in 2023 where they aligned 240 mammalian genomes and found that a third of these regions in our genome are actually actively evolving. And it was more interesting to know that most of these regions, they are involved in development of the brain. So my first postdoctoral work involved identifying these regions that are under high turnover because of these population variants. So what I did, I created two artificial genomes by substituting our reference genome with all the mutations that have single nucleotide mutations that are present in the chimpanzee at the corresponding position, as well as variants that are found in 1,000 genomes catalog. So with these, I found that there are certain regions associated with especially these genes that undergo high turnover. Interesting thing about these genes is that most of these are associated with disorders like autism, ADHD, bipolar, etc. So the understanding, and this comes from other studies as well, is that those variants that led to development of human cognition also led to recurrent neurodevelopmental diseases. So, but these regulatory variants can have a broader impact. They can alter the epigenomic landscape of a cell type, multiple cell types. So, for example, this is a differential methylation profile in of autism patients. This is hyper ASD, hypermethylated regions, and hypomethylated regions, and across different neuronal cell types. These are different cell types of the neurons in the brain, prefrontal cortex. And these hyper or hypomethylated regions in ASD, they're associated with, ASD is autism spectrum disorder. These are associated with some of the transcription factors that are quite crucial in the development of autism, like NFE2, June force, etc. So now, when you have variants from Jivas, this is typically how a Jiva summary statistic looks like. This is a set of variants that from a very recent Jiva study of ADHD from European cohorts. You have variants here, their corresponding genomic positions, and then the alleles, A1, A2. So here you have odds ratio. Odds ratio means the risk of developing that disease with this allele A1. Now, if it's more than 1, it means it's a risk allele. That means the G allele at this position increases the risk of ADHD. If it is less than 1, it means that it's decreasing the risk of, this G allele at this position is decreasing the risk of ADHD. It's called protective allele. And it's very important to know these protective alleles because they can help in developing therapeutic strategies for targeting ADHD or other phenotype. Okay, so another thing about Jiva studies is that they usually perform genotype microarrays. And I'm not talking about the UK Biobank, that's quite comprehensive, but other Jiva studies, they typically detect 500,000 to 1 million SNPs. But we have many more than that. We have variants that are much more than that. So these studies rely on something called the LD structure, linkage disequilibrium. These are regions of our genome that where the recombination is quite high. And so if you have a tag SNP, these are tag SNPs identified in Jiva studies, you can pick variants where, so the signal might be coming from a locus, but this may not be the causal variant. There can be variants which are in strong LD with these variants that have an indirect association. So we usually expand our list by including the tag SNPs and those in very strong LD to obtain a final list of potential disease associated variants. And then there are many fine mapping strategies to identify causal variants from these set of variants and then and their target genes. Another point about these variants is that they can have varying levels of disease penetrance. Now, this is a very popular plot in Jiva's community. This compares the effect size of a variant, that is how significantly a variant is associated with disease risk, to the allele frequency of that variant. So here on one spectrum, you have variants that have a very large effect size and they're quite rare. It means that if you have that mutation, mostly these are coding mutations. If you have this mutation, you are highly likely to develop that disease. For example, cystic fibrosis. Or another example is that of loss of function variants in a gene called MC4R. If you have mutations in this gene, you are highly likely to develop obesity, some form of obesity. Now, but these are quite easy cases to target because you already know the variant, coding variant, you know the gene, and you can target therapies. I mean, gene editing is editing usually targets such genes with monogenic forms of disorder. For obesity arising from MC4R mutations, currently there is a drug called setmelanotide that alters the levels of this protein expression to restore the physiologically normal levels of protein in, I think, the hypothalamus. Anyway, so but then on the other end of the spectrum, you have common variants with very small effect sizes. Now, most of the variants that you uncover from Jiva studies, they are actually common variants with very small effect sizes, and it's actually the aggregated effect of all of these variants in an individual that leads to disease phenotype. But and these can be obesity as well. For example, this is a Jiva summary statistic from obesity, the recent obesity study, Jiva study, where MC4R is a risk locus, but then variants associated with MC4R actually lie in the non-coding region. So that's a different effort to identify which non-coding variant is responsible for disease levels of MC4R, for example. So a disease can be can have a monogenic form, or most of the diseases can have a monogenic form or highly penetrant forms, and they can also be polygenic disorders. And then there are these common variants with very large effects, like ApoE4 variant of Alzheimer's, but that's not a topic for today. So once you have a Jiva summary statistics with odds ratios, you can calculate something called polygenic risk score. You can aggregate the effects of variants to predict the phenotypes or a risk, perform a risk assessment of a patient. You have, for example, here for a cardiovascular disease, you have different loci that lead to cardiovascular disease phenotype, and then based on certain thresholds that are obtained through statistical analysis, you can predict the disease risk. But these studies are not going to tell you how to develop therapeutic study. I mean, obviously, you uncover some risk loci that can help you understand which regions are involved in disease risk. But then if you want to move towards therapeutic strategies, you need to understand how each of these variants are involved in a mechanism that leads to disease phenotype. So first of all, and I'm going to mostly talk about regulatory variants here, non-coding variants, because for coding variants, as I said, it's quite easy. If there's a missense variant in a gene, you can target that gene to restore its normal levels through CRISPR and whatnot. But then if you have, so for regulatory variants, the challenge is that you first need to understand from which cell type the variant's effect is emerging, manifesting. And then you need to identify, if you have 100 variants from which the signal came in Jiva study, only a few of them would be actually functional. So to do that, there have been very popular studies called massively parallel reporter assays, where you transfect plasmids with uh, you can have multiple variants at the same time, transfect cell types with these plasmids of your or variant of interest, and then you count the DNA to RNA ratio, and that tells you if that variant has a regulatory activity. Or the most state-of-the-art approach right now is crop-seq, that couples CRISPR with the genetic with single-cell RNA sequencing. This is a very specific, precise approach to identify which variant in which cell type is leading to a disease phenotype by directly measuring RNA-seq levels that are affected through that variant. Now, if you have all the time and money in the world, you can perform these experiments across more than 400 cell types. And I told you there are like millions of variants. And then there are 1.4 million regulatory elements that have been cataloged by ENCODE so far. But then that's not feasible. I mean, there will be more variants that will come up. There are so many rare, you don't know which one to pick up. So in the past decade, the most popular approaches have been deep learning models. And this is my field as well. I work on deep learning models to uncover functional aspects of regulatory variants that are associated with the disease. So back in 2015, it started with the predecessor of a model called SAY. It was called DeepC. These models were mostly convolutional neural network frameworks. But then most recently, you have hybrid models like Informer and Alpha Genome, which combines the convolutional neural networks with transformer architecture. And just a brief introduction about what these models do, how these are trained. So these are epigenomic profiles from a cell type. The reads here, which represent active enhancers or open chromatin regions, they are converted to one hot encoding that looks like this. And then there are several convolutional layers that extract patterns out of these sequences. And then there will be combinations of these patterns. So that's why you have multiple convolutional layers, eventually giving you an output of patterns that represent the input sequence, which you provided for either HTK27AC as active enhancers or open chromatin regions. So, and then it will predict a profile that looks like this. But the disadvantage of using CNNs in the first place was that it was localized to either 2 kb or maximum to 20,000 base pairs in the genome, which is very local and small when you compare enhancers, because enhancers can sometimes act from like 1 million base pairs away. So you want these distal interactions to be accounted for when you are training a model. So that is when I think Informer or DNA Bird started incorporating transformer layers in these models. What that does is, it for after you have these convolutional layers, it will identify the influence in one genomic region of another distal genomic region. So how this position is influenced by this position and so on, and it can do extend this attention layer up to 1 million base pairs. And once you have this information, you can output gene expression by predicting distal enhancer interactions as well. So this is not all just useless. Okay, but before that, then how you can incorporate variant information. You can perform in silico mutagenesis on your input sequence and identify how the prediction changes. You will have some profile called contribution score of different bases, and once you have that, you can identify which of these variants may have more impact on gene expression. So, like I said, it sometimes most of these models actually have a very good correlation with experimental assays. You can read the Alpha Genome or Informer paper. They have validated those scores that they get from the model with experimental assays. But here, this is a correlation from our lab using an in-house deep learning model that's based on CNN architecture. They correlate the activity obtained from MPRA assays to activity predicted by the model, and it's pretty decent. The best you can have from computational techniques. You can at least narrow your candidate list down to a few that can be tested using experimental assays, right? So once you have your set of causal regulatory variants, either predicted or from assays, then you want to find out what are the cognate cell types in which a disease manifests. So I'm going to illustrate that using one of the very comprehensive studies done so far for autism. So they had single cell ATAC-seq and single cell RNA-seq from the frontal cortex of the human brain. They have cell clusters. These are divided into two different types of neurons: inhibitory and excitatory neurons, microglia, astrocytes, etc. Then they have GWAS from autism. They get the causal variants from that set, and then they look for enrichment of those variants in these cell types. Now they began with data from frontal cortex because after all of these studies so far, it's a general understanding that neurodevelopmental disorders are mostly localized to neurons of the brain, neurons of microglia, astrocyte, etc. So they performed a study only in the prefrontal cortex. They found that variants associated with ASD are enriched in microglia and the neurons. So then you have a few state-of-the-art tools. Here, I'm depicting a test case for scenic. So they correlate the single cell ATAC-seq, where the variants lie, with RNA-seq to perform to identify regulons or these gene regulatory networks using co-expression profiles. And these networks, for example, here in microglia in autism, IRF8, which is a transcription factor, is responsible for forms a regulon that regulates gene expression in microglia, and CUX1 in neurons. So you have identified these central transcription factors using single cell ATAC-seq and RNA-seq data. Once you have these regulons, you can then go to perturbation atlases. Here it's depicted using a database called LINKS. So what these databases contain are perturbation of small molecules or transcription factors, and how that perturbation may have an impact on different cell types. And they have differential gene expression profiles. So if you have a regulon, then you can look into what the effect of perturbing IRF8 in microglia would be using these databases. And it also tells you about agonists or current drugs that perform the same give you the same differential gene expression as IRF8. And then you can use that as a potential therapeutic strategy against autism. So these perturbation cell atlases may have varied applications, and not just this. You can have these cell atlas perturbation data from one cell type, but then you can also test whether that perturbation may have a different kind of effect in other cell type. So that will give you an understanding of how precise your approach using one type of drug can be. But I am more passionate about this other research that recently happened. That the study is still in preprint. What happened here? They targeted the cell type specific expression of a gene called SCN2A. This gene is associated with autism, and it's downregulated in neurons. So they wanted to see if they could restore the expression of SC2NA in autism patients. But so that for that they use cortical organoids, and using CRISPR activation through a brain enhancer, a fetal brain enhancer. So what the property of this enhancer is that it's not active across other cell types, it's active in fetal brain. So if you target variants, if you use these fetal brain enhancers, you can modulate bases that can lead to upregulation of this gene in a particular cell type, in this case, neurons. And this is what they did. In wild type neurons, which do not contain any mutation, there is no change in gene expression. But in those cell types which have this mutation, haploinsufficient, this CRISPR activation led to upregulation of this gene. And they observed that in this cortical organoids, which are cell systems, not the endogenous context, increased expression of this gene led to normal expression of this gene in upper layer neurons and neuronal firing. That I think is a very exciting direction right now. So the challenge of identifying regulatory regions, their cell type specificity here has been leveraged as an opportunity for targeted therapies. I think the study is still under review. But anyway, aside from talking about therapeutic strategies, I thought it would also be interesting to discuss where these studies can also take you in terms of evolutionary mechanisms that are shaping our regulatory landscape. So I spoke about loss of function in intolerant genes. These are genes that are depleted of mutations, but in my current study, I observed that these genes are enriched for diseases associated with development, particularly neurodevelopment, like developmental delays, anemia, intellectual development disorder, etc. And the effect sizes of these variants from whole exome sequencing seems to be much larger for these set of genes compared to the other set of genes. And I also found that the enhancers of these genes, I call them essential genes, they are enriched for diseases like age-associated disorders like Alzheimer's, Parkinson's, and vascular dementia, etc., and as opposed to other genes. So, and also they harbor rare variants with very large effect sizes, which leads me to, it means that these genes, these loss of function intolerant genes, may have a very biased effect on human diseases. So my current work involves understanding how this might be. I'm using a combination of deep learning models and other expression markers to understand how these genes are contributing to these age-associated dementias. But so, in addition, I think it's also very important to point out why we need data from other cohorts. As I mentioned earlier, UK Biobank contains a large cohort of European population with 7,000 phenotypes, but other populations are underrepresented. It's coming up, but I just wanted to highlight why it's important to have more data at that scale from different populations. So this is a comparison of jivas from different traits like tobacco use, arthritis, etc. And there are variants associated with this jivas, and their frequency in American indigenous ancestry, which is quite low, but it can be quite common in European ancestry, and so on and so forth for different loci. So this means that first of all, the disease prevalence cannot be extrapolated from European population studies to other populations. Second of all, it can be it's possible that the pharmacogenetic variants that you identified from European population can be totally absent from other populations, or there can be other variant in other ethnicity like Hispanic or Indian that is leading to the disease from the same locus, but not the variant that was identified from the European ancestry, which means if that is the case, then that variant might be involved in some other kind of mechanism altogether to mediate that phenotype. And that's why we need to have more a deeper understanding of population specific mechanisms. But why are these frequencies so different? So for that, I thought I'll explain through a couple of slides. I think 2 or 3 more slides left. So this particular study, to understand how evolutionary history has proceeded with human environment. Obviously, these allelic frequencies have changed because of environmental factors. So there was a study that aggregated data from 230 ancient individuals between 6,500 to 300 BC from present-day Europe, and compared that with present-day European population. So these are the ancient populations, these are the current populations, and two examples here. So this particular variant, it in the gene SLC45A2, it contributes to light skin pigmentation, but it was present in very low frequencies in the earlier ancient population, but has risen up to compare. I mean, the present day human population, European population has a very high frequency of this allele that leads to lighter skin pigmentation. And then there's another variant that in that West Eurasian European population, they relied mostly on agricultural diets. So there was a mutation that protected them against ergothioneine deficiency. So this was a deficiency associated with agricultural diets, but that led to prevalence of celiac disease. This is how different environmental factors lead to population-specific traits. But one of the most interesting aspects of this allelic heterogeneity was, I think, in my opinion, is this about this particular variant. This is a missense variant in a protein called TIC2. I think so far this is one of the very few variants that protects against 10 different autoimmune conditions, including psoriasis, multiple sclerosis, Crohn's disease, etc. The odds ratio is less than 1 here. But in European population, this variant is present at very low frequencies. I mean, it was at a higher frequency in Bronze Age and Iron Age, but then it decreased. And the decrease actually corresponds to the age when TB was endemic in Europe. So what happened was this variant provides a predisposition, monogenic predisposition to TB because of disruption of IL-23 dependent antimicrobial IF and gamma immunity. Now, this case provides a pharmacogenetic warning. If there are drugs that have been developed to inhibit TIC2 protein based on the protective effect of this allele, they can potentially lead to increase of infections due to TB or involving TB or other mycobacterial diseases. So that led me to, I mean, I thought I'll conclude with the slide that it's very important for all of these reasons to have whole genomic sequencing data from India and to a very large scale. Currently, the Genome India project has WGS data from 10,000 individuals, and I believe the trait and disease and insights are still under analysis. But I think once we start having larger data from larger cohorts, like haystack analytics is, I think, starting to do, it will be very useful to identify therapeutic strategies to tackle obesity, neurodevelopmental disorders, diabetes, etc., which is becoming quite prevalent in the Indian subcontinent. And once you have this, you know your data, you can start turning, as NIH logo says, turning discovery to health. With that, I conclude, and I hope there are any questions I can answer.
Thank you, Jaya, for the wonderful talk. I think the way you covered, right from the databases that are present, the amount of data that is out there freely available, and then to connecting it to the work that you are doing, I think it was a fantastic effort.